companies presenting, and also 4 companies at 4 different stages of fundraising and commercialization. So quickly about myself, my name is Eric Wong. I'm the co-founder and CEO of GaudiBio. GaudiBio is named after the Latin prefix for gaudium, which stands for joy. And all we're trying to do is to build the best preclinical model that can predict clinical trial outcomes. So with that, I wanted to just quickly go over our agenda. I know we're already late, so I want to get going with Bryan. Bryan will talk about advances in preclinical, specifically for GBM, or glioblastoma, and then Bhaskar, who is the CEO of Cordance Medical, will talk about a non-invasive way of delivering by opening up the blood-brain barrier using focal ultrasound. Take a quick break, and during that break, take some of you in for a tour of our lab space, see where some of these brain organoids are being made. And you can, I don't know, depending on how Andrea feels, because you do have the gown up, so maybe we can take 2 batch of cohorts of people. And then after we come back, Mike will share with us his company's experience with eradicating the root cause of GBM and cancer, which is very, very exciting. They're in a preclinical stage, and then we're going to cap it off with Tracy's talk, and she's going to talk about human data. So we have 2 therapeutics company, preclinical company, and a delivery company. So all very exciting. Very quickly about GaudiBio, this is our mission. Wanted to address that 80% of the CNS drug clinical trial failures by addressing efficacy and toxicity, these 2 things that account for the majority of the failures. Do that by creating these cohorts that are living in petri dish with good representation of the intended use population and its various subtypes. As you know, whether it's cancer or other diseases, it's not just one disease, it's really a syndrome. And we do that and use different modalities. I would just stop there and just tell people that since the 2 months that we've been in operation, made 10,000 organoids. Wow. And the power of having organoids is an ex vivo experience where you can track things longitudinally, non-destructively, so you can actually see longitudinally how these organoids are behaving, have electrical activities and physiology that you can measure outside the body. And just, it's all about access. With that, I just want to say thanks to everybody and quote this, a quote almost as close to JFK's quote about going to the moon. The use of human relevant models will go ahead, whether we choose to embrace it or not. So thanks, everybody. I'll hand it over to Bryan. Bryan, do you want to present on your own desktop, or how do you want to do it? Or you want me to bring it up?
However.
And also thanks to CSD, of course, and the Stanford Stem Cell Institute for hosting us and providing the valuable space, resources that's available to us. Sorry, Bryan, go ahead.
Whichever is easier for you. If it's easier just to present there, that's fine. If you want me to, I'm happy to do that too.
Oh, okay. I can bring up your slide. 1 second here. Oh, okay. I stopped sharing, didn't I?
So while he's pulling up the slides, I just want to say hi to everyone and I guess share a story. In a past life, I was working with another company, and I had mentors tell me all along that we've cured cancer in mice over and over again, but it's not so easy in humans. And I was leading well, I wasn't leading, I was working on a program where we were working on a project. It was an IL-2 program, and we were seeing amazing data. We were collecting the tumor microenvironment, and we were trying to figure out how our immune oncology agent was working. And we were working with a colorectal cancer line and just having amazing results. And I was sure this was it, and I would have bet the farm on it. And it went into the clinic, and and it just didn't do anything above baseline, and it was so disheartening. And all along, I've been more heavily on the neuro side for most of my career, and I've been banging the drum that if you really want to understand human biology, you need a human system. That's where it's at. So so to introduce myself, I'm an organoid zealot and the head of biology at GaudiBio. Next slide.
Oh, sorry.
So probably everybody on this call is already aware, but of course, glioblastoma is rare but devastating. That even though there's, I say, only 12,000 cases per year, it often hits people in the prime of their life from the 40s to their 60s, and median survival is really terrible, 15 months, and fewer than 7% can survive for 5 years. And it blows my mind that there's been no true drug advances since TMZ in 2005. And a quote that resonated with me, and I read this as I was learning about glioblastoma, is that it steals people's identities. And a lot of family members say, I lost my love twice, once to the disease and then once to death. And that's because this attacks your brain, and that's what makes us human, the memory, speech, personality. And that it really struck me in learning about glioblastoma. So that's right, 20 years, hundreds of clinical trials, and one drug, TMZ. That's the only drug that's improved survival. Why? Preclinical models can't reflect the human brain or the tumor microenvironment, and mice are not people. Animal success does not, just like my experience with nemvaleukin, mice are not people, and animal success doesn't translate to patients. But organoids change the game. They're entirely human. They preserve tumor heterogeneity, microenvironment, and neuron-tumor interactions. Furthermore, they can be patient-specific, and they provide a path to personalized therapy. So for in vivo models for GBM, I'm not going to read through the table to the left, but just suffice it to say that the pros of rodent models is that you have the whole organism complexity, you have a systemic immune response, but the cons, and I can't understate this enough, or say this loudly enough, the species difference, cost, ethical issues, and the FDA push to reduce animal use. And bottom line, rodent models fail to predict clinical efficacy. So for some of the in vitro human models, in 1981, that marked the beginning of the conventional GBM cell lines that are in use. And here's a table showing biological relevance and throughput and cost. And you can move from left to right as the models get more and more sophisticated, that from conventional cell lines, we can move to early passage, patient-derived lines, 3D scaffolds, tissue-sliced cultures, and then, of course, my personal favorite, GBM organoids. And then there are more sophisticated models that have come out more recently, tumor on a chip and bioprinted GBM models. But we feel that the sweet spot is really at that organoids. Why? Because you have high complexity. I take issue with the throughput. I think we can improve throughput, and the cost isn't as high as it is with some of the other models. And the pros are that these are human-derived, higher throughput. They recapitulate the human tumor microenvironment. Some of the limitations are the limited vascularization and an incomplete immune components. But for both of those points, the pros and the limitations, I want to remind everyone we're still very much in the early days of making these organoids, and I think there's a lot of room for improvement. So just to touch on the FDA Modernization Act and the regulatory landscape, I think we all are familiar with the fact that the FDA is no longer going to require animal testing before human trials. And I think that's going to lead to an increased interest in these human in vitro models and a potential acceleration of these models. But why would the FDA be interested in making this switch? And I think it's because if you take a look at the way drugs have historically been delivered, is through the use of cell lines and inbred mice. And if you look at the genetic diversity within these sorts of systems, they're very homogeneous. There's very little diversity. Whereas when you go to a true clinical cohort, now you have a much larger diversity in genes, and then that leads to responders and non-responders. And this is a truly human experience that we need to model earlier. So if we take a step back and we think about, I think it was 2011, Hanahan and Weinberg listed the 11 hallmarks of cancer. And if we take a look at 2D cell lines, animal models, and organoids, we can see that animal models and organoids are pretty similar in being able to recapitulate a lot of the hallmarks of cancer. Click forward, please. But if you take into account the hallmarks of glioblastoma, including the heterogeneity of the tumor, the stemness and plasticity of glioblastoma, the neuron-tumor interactions, the immunosuppressive environment, these are places where, yeah, animal models and mice have all those things, but they're not human. And the differences between human and animal make a difference in the activity and function of the drug. Next slide. So why are human systems better? Well, first, species-specific regulation. Yeah, human and mice both have transcription factors and OCT4 and ANOG, which are so crucial to glioblastoma. They behave differently in humans and mice. They're regulated differently. The epigenetics around OCT4 and ANOG are different. The downstream signaling of these genes is different. Context dependent. Hypoxia, ECM, neural niche signals. So things like humans have more glia than mice do. All of that leads to the stiffness of the ECM, and the stiffness of the ECM leads to gene expression changes. So translational fidelity, human-specific drug responses tested in human will lead to fewer than mouse to human failures. And then just ethical regulatory climate. The FDA is shifting towards human and non-animal models. So I'm going to talk about two types of organoids. I'm going to talk about glioblastoma organoids, which I'll refer to as GBOs, and glioblastoma cortical organoids, or glycos. What's the difference between a GBO and a glyco? Well, first with origin. GBOs are entirely derived from patient tumor fragments, whereas glycos are cortical organoids with GBMs sprinkled in. What are they best to model? GBOs are better for tumor heterogeneity, epigenetic, and genetic changes, whereas with glycos, you'll see that you can actually model tumor invasion and infiltration, and you can definitely see neuron-tumor interactions. Some of the advantages of GBOs is that you can make them very quickly. They're higher throughput, and you could relatively quickly do personalized drug testing on glioblastoma patients, whereas glycos are better for looking at tumor-brain crosstalk. You can use functional readouts like MEA and calcium imaging to take a look at the organoid, and it also captures the invasion dynamics. GBOs are limited because they have essentially no brain microarchitecture, and they lose stromal cells with additional passaging. Glycos, on the other hand, take longer to set up, and they're technically complex, so they're lower throughput because of that. GBOs are particularly good for drug screening, looking at genetic or epigenetic studies, and fast patient-specific testing. Glycos really are the thing to use when you're looking at mechanistic studies, if you're looking at invasion circuits, network activity assays, and tumor niche interactions. And I'm going to focus on these two references that are below. If you just have time to read two papers and you want to know most of what's going on in this field, these are the papers, and we're going to take deeper dives into each of them. The first one being GBOs. So this is a Cell paper. At this point, it's a few years old, so apologies if you're already well familiar with it, but I'd like to walk you through this. Next slide. So first, how do you make these? Well, they're relatively straightforward to make, actually. You isolate a glioblastoma tumor, and then you essentially cut it up, cut it into pieces, and then culture it. And within 2 to 4 weeks, you start growing these GBOs. And then as they grow larger and larger, you can cut them into smaller pieces to avoid necrosis. And the good thing with these is you can freeze them and biobank them. So the timeline for that is about 2 to 4 weeks. Next slide. And GBOs have characteristics similar to GBM tumors. On the left panel C there, you see CD31. This is a marker of microvascularization. So we're getting vascularization within the GBO. And on the right, what you can see is that heterogeneity that you want to capture. At the center of the GBO is a necrotic core. And then as we walk out from the core, you see a region that's turning magenta. That magenta is a hydroxy probe, and it's showing hypoxic point. And you see fewer green cells. The green cells are a Ki67 probe that mark proliferation. There's reduced proliferation because of the hypoxia. And that hypoxia leads to cells that become senescent. And that's where a lot of the resistant cells are to chemotherapy and radiation. The outer portion, you see more green cells. That's because that's the more proliferative zone. And that's where you find your GSCs, your stem cells are in this area and are more highly proliferative. Next slide. So in addition to seeing some of that heterogeneity, I know the slide looks busy, and I just want to walk you through that. On the left is one tumor, the 7966, and on the right is a second tumor, 8036. And what I'm trying to show in this slide is that they look similar. If you compare the tumor to the GBO that's right next to it, that's a 2-week time point of the glioblastoma organoid, and then to the far right is the biobanked version. So it was grown for an additional 2 weeks. And you can see even visually that the cell structure looks remarkably similar, that the same sorts of cell types. So at the top with GFAP and S100 beta, these are our astrocyte markers. And in the middle panel, we're seeing nestin and BLBP. These are our glioblastoma stem cell markers, and we're seeing the same occurrence of those. And same with SOX2 and OLIG2. These are markers of stemness. And for a more quantitative view, looking at panel B, you can see that across SOX2 positive cells and OLIG2 and Ki67, that even after weeks in culture, the relative population of the cell types reflect the parental tumor. So you can keep these for up to 48 weeks without deterioration of these signals. Next slide. I'll go quickly over this, but this is just showing that over 90% of the tumors can be created as organoids frozen and thawed. And this is just what they look like coming out of thaw. And yes, some grow faster than others. So you see faster growth curves for some than others. But this reflects the heterogeneity of glioblastoma. More importantly is the gene expression. So what we're taking a look at here on the left is about a dozen different tumors. And we're comparing the gene expression of the 10,000 most variable genes to organoids at 1 week, 2 weeks, 4 weeks, and 12 weeks. And you can see that the tumor gene expression compared to the organoids at the different time points doesn't change. The gene expression is staying the same within that cell line. Panel B, on the other hand, to the right, we're looking at the same cell lines. And just because the cell lines keep expression within within that tumor, the expression is different from tumor to tumor. So it's well understood that glioblastoma is not just a single flavor of glioblastoma, but there can be very many variants with different gene expression profiles. And what I'm showing here in panel B is that over time, the gene expression doesn't change for each cell line. So we're maintaining heterogeneity across different glioblastomas, but maintaining the gene expression pattern within each cell line over multiple passages. So that was bulk sequencing. They've also tested this by single cell sequencing. So I thought this was really interesting when I first read this. This is taking a look at 4 different tumors. And what they did was when they did single cell, they found that each tumor, so that the data that's shown here is the parental tumor and the GBO sequenced together. And what they find is that the GBO and parental cells cluster together, but they cluster separately from each other. That's what you see in panel B, that each of the tumors and GBOs cluster separately. What you see in panel A is that the cells that came along for the ride, the macrophages, the microglia, oligodendrocytes, and T cells and stromal cells, they all clustered together from all the different tumors. So even though they came from different tumors, it's kind of an internal control. You can see that the oligodendrocytes are still oligodendrocytes despite the patient, but the tumors can vary. Next slide. And then on this slide, we're taking a look at the macrophages in the immune system. And what's interesting is that the macrophages look similar in tumor and GBO, and that their cytokine profile doesn't change. That the same cytokines that they were secreting in the tumor, they're still secreting as GBO. What is also interesting is on the right is that tumors have more microglia-like content. So the myeloid cells tend to be more microglia-like. And as an organoid, they shift to more macrophage-like gene profile. And this is one limitation of the GBOs, that while the cancer cells are proliferating, the myeloid cells are not. So every time you passage these, and as they grow bigger and bigger, you're shrinking that immune content. This single cell sequence on the left, panel F, we're taking a look at a tumor now, and the organoid at 2 weeks and 8 weeks. And what we can see is that there's no major shift. The cells stay roughly with the same distribution over weeks as an organoid. And on the right, in panel G, we're looking at the tumor compared to the organoid at 2 weeks. And then taking a look at the different clusters from the tumor and the 2 week. Now, there are some differences, but by and large, at the bottom, if we take a look at the Pearson correlation, along the vertical axis are tumor clusters, and the horizontal axis is the GBO clusters. And we see good correlation between the high expressing genes and then the low expressing genes from these different clusters over time. So they don't change. But how do they xenograph? And we see robust engraftment into mice. So panel A is an MRI signal, and glioblastoma responds with a flare signal, and the flare signal can be seen in the glioblastoma for the 2 different cell lines, and as well as H&E staining on the right in panel B that across 2 different tumors, looking at the tumor and then the xenograph, we see similar architecture and similar morphology from the original tumor to the xenograph. And this is also true with ICC if we take a look at staining. So these are 2 different tumors, the 7788 and the 7790. These were xenographs that were put in, and in the pink one, box 1 shows where the xenograph was originally put, and then box 2 shows where they sampled as, do the cells change as they proliferate deeper and deeper as they move from box 1 to box 2. And if you move to the right, if you take a look at the staining, you can see box 1 is like the top line, and then box 2 is the bottom line. As we move from left to right, and Doglin is a marker of vasculature, we can see that they're vascularizing in both local and distal spots. STEM 121, SOX2, and Nestin are all glioblastoma stem cells, and we see those are maintained. And then EGFR V3 is a variant in some glioblastomas, but not others. 7788 had the EGFR V3. The 7790, or the bottom one, did not. And what's encouraging here is to see that it didn't pick it up in 7790, nor did it lose it in 7788, which has been known to happen with other samples. So it maintained that differentiated EGFR expression. This was the data that really kind of most excited me. So here on the left, we're taking a look at 4 different patients, 7790, 803, 7788, and 7884. These are tumors that were taken and converted to GBOs. And then they were given the standard of care. The GBOs were modeled with the standard of care treatment with 10 gray radiation and then TMZ. And what you're looking at is the gray cells in the top and bottom. And you can see that with 7790, there's very little difference. Like by eye, it looks like you see as many white cells in the top and bottom. Similar with 7803, maybe it looks like there's a few fewer on the bottom. But 7788 and 7884, you can see that there's a big reduction in the white cells in the bottom. As it turns out, and I hate to say this, but the 7790 patient fared very poorly. They only lasted 1 month beyond treatment. 7803 only lasted 3 months beyond treatment, whereas the other two patients excelled at the median standard of care. So on the right, you can see there's additional data and quantitation. One thing that the authors could not explain is the MGMT methylation. It's believed that MGMT methylation should be predictive of treatment response. However, they did not see that. Can point out that the authors were very conservative with this result and said, this isn't statistically significant. We can't say, I said it, but they wouldn't say it. They wouldn't say GBOs recapitulate patient response. They said, this looks encouraging and we should do further testing. But I thought, wow, that's really exciting. So that's my editorializing, not theirs.
Okay, Brian, you're at the 10 more minutes.
Okay. So here's an example of CAR T treatment on these GBOs. And to walk you through this experiment, there are 6 different tumors that you can see from left to right there. And I wrote low EGFR-V3 or high EGFR-V3. So these tumors are either low, high EGFR-V3. And then they were treated with 2 different types of CARs. CD19 is a negative control CAR T treatment, and then 2173 is an anti-EGFR-V3 variant. So now to step you through from top to bottom, CD3 intensity is a T cell marker, and that's showing if we get T cell proliferation. And we can see that all of the cells showed some proliferation, but the high EGFR cells proliferated even more. CCC that there was higher amounts of apoptosis wherever there was a CAR T with the EGFR-V3. And then the bottom one shows that they destroyed the EGFR-V3 cells. So this was very encouraging to show that an immune therapy could be modeled in vitro in the system. However, the CAR T did not kill all the tumor cells, and they persisted after 3 days. The authors speculate that there were probably some glioblastoma cells that were EGFR-V3 negative, but they were still glioblastoma that escaped treatment. So with that, I'm going to turn the page to the other type of organoid, which is called a glico. And this is from the Howard Fine lab at Weill Cornell Medical Institute in New York City. So this is a very, very nice paper. So just as an overview, how do you make glico organoids? They use a different organoid protocol than we do at Gaudi, but it's similar. Basically, they make cortical organoids, and then from those cortical organoids, they get rosettes, and then they embed them into matrigel, leading to, they call them mini brains or organoids. And then you seed them with glioma stem cells, and then you form a glico. And this glico is a co-culture, essentially, of the glioblastoma cells and the organoids. Well, what do these things look like? Well, after 1 week of growth, you get what's on A. Oh, I should have mentioned the glioblastoma cells, they label with a lentivirus GFP, so you can see them in the culture. So anything that's green, you know, is GFP. And one of the things that's encouraging in panel B is that you can see consistency. So the top and bottom samples, 7 to 8, tended to grow very quickly. And in the glycos, those were the most aggressive. So they were very invasive and grew rapidly. The next to 320 and the bottom next to bottom 810, those grew as balls, like neurospheres of cells. And when they grew, they developed a honeycomb pattern in the organoid. 517 and 607, those two in the middle, those were the slowest growing glioblastoma in 2D. And when they put them in the glico, they saw that they were very splotchy, and they developed a different sort of invasiveness than the others. And one of the things that a hallmark of glioblastoma are tumor microtubules. They're unique to glioblastoma and other aggressive brain tumors. They allow for communication between cells. They support tumor invasion, and it's believed that they help protect the organoid. That when the glioblastoma is undergoing treatment, it steals what it needs from the healthy cells. So people have observed mitochondria passing from healthy cells to the tumor cells. And when the glioblastoma moves in, all the surrounding cells die. And it's believed that this could be one of the mechanisms. So in panel A, you're seeing a 2D culture and what these microtubules look like. So that's just a 2D culture of glioblastoma stem cells. Panel B is showing the GFP stained glico, and you can still detect them. Panel F is a time-resolved image, and when they do calcium signaling, they actually can see voltages passing through these neighboring cells, one stained with GFP and the other with RFP, and it looks orange because you can see that there's crosstalk between the two of them. So the fact that it preserves these microtubules is very important. So glico models are also resistant to chemotherapy. So on the left, you can see that both TMZ and BCNU both knock the heck out of glioblastoma cells in 2D. We see an 80% reduction in TMZ and a 90% reduction with BCNU at the highest concentration. But over on the right, you can see that BCNU just tickles it. It doesn't even, 923, I mean, that in the bottom, TMZ knocks 923 down by about half, but BCNU really doesn't do anything to it. So you see major differences in what chemotherapy does. And this is even more highlighted in the next slide I have on the left, is how 2D cultures respond to radiation. And you can see with 5 gray, you get a 44% reduction, and with 10 gray, you get a 74% reduction in cell viability. But in the glico, 5 gray, really there's no perceptible change, and that's really visual. You can see that visually. And with the 10 gray, there's about a 25% reduction compared to 0. But you can see that the cells are much more resistant to the radiation in this condition. This isn't so interesting, but glicos can grow equally well on 1-month-old organoids or 4-month-old organoids, and ESCs, or embryonic stem cells, or iPSCs, that they like both scaffolds, and they grow equally well under both conditions. So this is just, I hope that you found this helpful and informative, and I really just touched the tip of the iceberg, but there's a lot of applications for these 3D organoids, and I think this slide captures a lot of them that I touched on. So cancer modeling, being able to see the invasiveness of glioblastoma within the cerebral organoid, and then I'm just going to work my way clockwise. Drug screening, being able to use particularly the GBOs to quickly develop treatments, and as we move down, personalized treatments. You can even test different drugs on a person's resected tumor. CAR T treatment. I didn't have time to talk about it, but there was a paper that came out this year that mixes a person's PBMCs with their own tumor to look at things like CAR T treatments and cell therapy applications. Genetic engineering, I didn't talk about that at all, but you can certainly use the system that way. And I showed you some examples of transplantation, of being able to do xenografts for these back into mice. So in the past, so from failure to future, and organoids matter. So we could keep doing business like we did in the past, where there was over 1,000 clinical trials with one drug, or on the right, we have a human-based, functional, predictive, and personalized approach. And next one. Animal models fail patients, but human organoids bring us closer to cures. Thank you very much. I hope that wasn't too fast, but there was a lot I wanted to say, and I was passionate about it.
Thank you. Thank you, Bryan. I know, given our time constraint, perhaps we could move on to our next speaker, and then we could perhaps follow up with Bryan with additional questions. So next we have Bhaskar Ramamurthy, and he's going to tell us about the innovative technology that Cordance Medical had developed on focal ultrasound and how it can open up the blood-brain barrier for very targeted delivery. Okay, so let's see. Bhaskar, do you want to present on your end, or you want me to project?
Yeah, no, I can present from my end. It's a little bit easier, Eric. Yeah.
Okay. All right.
So can you give me permissions to share my screen, please?
Yeah.
Good. Okay. Focus back on GBM because it's application. So we'll talk about that. We do have the FDA breakthrough device designation, which allows us to talk to the FDA very frequently, and they give us really good guidance on how to get through the regulatory processes. So again, my name is Bhaskar. I'm the founder of this company, and as a background, I'm a biomedical device engineer. And I'm very fond to say that if any of you have had the ultrasound exam in the past, I've had something to do with that, because my technology has been out there since the 90s in almost all the ultrasound systems that are out there in the world. So I've been with ultrasound for a very long time in terms of how to use ultrasound for imaging. But this application is all about using ultrasound to get through the blood-brain barrier. The problem statement is very, very stark, and obviously all of you know that neurological diseases. I'm talking not just about brain cancer, but really all kinds of diseases. But I'll come back and focus around GBM here in a minute. So to me, when I look at the problems with treating brain diseases, it really comes down to two main issues. Of course, there are a few, definitely others, but it comes down to the blood-brain barrier. And for people who are not familiar with that blood-brain barrier, if you think about the brain has about 400 miles of vasculature, little vessels that make up the blood flow within the brain. And each of those vessels is covered with this really impenetrable layer of cells between the blood and the tissue itself. And this is very unlike any other between the blood and the tissue. Is very, it's quite easy. But in the brain, the transport of any particle, any drug particle, between the blood and the tissue is extremely hard. About 98% of the drugs, of the known drugs, do not cross the blood-brain barrier. And moreover, the transport between the tissue and the blood is also impeded, and that is really important in the context of diagnosis. Not a single monolithic layer. It's every single blood vessel has this layer, and if you want to visualize it, here's a small vessel, and these layers, these cells are very tightly interlinked to each other, and these are the cells that form the blood-brain barrier. It may be too basic for you, but I just wanted to put that out. The second reason it's really hard to treat brain diseases is of course, the brain is quite hard to access in terms of getting any pathology of the brain, and you have to go through the skull, which has its own risks and a risk of death and infection, whatnot. So when someone like me looks at the issues with in treating brain diseases, it comes down to the inability to cross molecules and drugs between the blood and the tissue, and the inability to access the brain easily. So this is a little bit more on that. In blowing up a small vessel here, look, we are able to go through that barrier. So here I'm showing a small vessel, and here are the cells that make up the barrier. And these cells are tightly interlinked to each other, as I just mentioned previously. And the diseased brain, the diseased tissue has biomarkers, and if you were to enter the blood flow, really good things can happen. I'll come back to that here in a minute. But biomarkers do not go from the tissue into the blood because of this tightness of this junction. On the flip side, when you have drugs that go into the systemic, circulation within the vessels, but they don't come out from the blood into the tissue itself, that's because these junctions are extremely tight. So this is a big problem in the blood-brain, in terms of blood-brain barrier, the inability to transport back and forth from the blood into the tissue. So what is a solution from a device standpoint? So focused ultrasound can actually open the blood-brain barrier very safely. This was discovered about 20 years ago out of Toronto, Canada, that with focused ultrasound, we can do a temporary and very local opening of the blood-brain barrier. However, the methods that people are using, that academics and researchers are using to get ultrasound through, requires very large, expensive machines, and require a huge set of staff that is, it makes this procedure very hard to communalize, to be able to, put out into the community. So the science actually exists to open the blood-brain barrier safely with focused ultrasound, but the methods of delivery are really hard for the patient, hard for the hospital system, hard for the clinic, and expensive. So, and therein lies, the invention that I made in 2016. So I started with the thought that, look, ultrasound can actually go through the skull, in a very unique way, and we can invent a non-invasive method that does not require a huge machine or anything like that to get to the skull, actually open the blood-brain barrier safely. So I took the existing science of opening the blood-brain barrier and invented a new science, a new way of depositing ultrasound through the skull with a non-invasive procedure. And that was the start of my company back in 2016. With that, I applied for 2 National Science Foundation awards, and I got that award, and the National NIH award. So all that helped me bootstrap my company with just an idea, with the 2 NSF awards, the NIH award, and then over time I've built a device that can open the blood-brain barrier in large animals, and we've got a demonstrable proof that we can open the blood-brain barrier in pigs, dogs, and sheep safely in a minute. So we've taken the existing science and invented a new way to deposit ultrasound. And here's what it is. So I'll come back and talk about the science of opening the blood-brain barrier here momentarily. But if you can imagine this, please, we have a patient here. The patient puts on a cap. This cap has multiple ultrasound sources, and with each sound energy going through the skull into the brain, and where all these ultrasound sources converge to a focal point, that and only there can we open the blood-brain barrier? So it's not a complete opening of the blood-brain barrier as you would have if a patient were to take mannitol. It's not at all like that. It's a very focal opening of the blood-brain barrier just around the area where the physician tells the device where to open the blood-brain barrier, which would be around the disease tissue, and the intermediate tissue hardly feels any ultrasound at all. So the bioeffects are just all focused, and that is what we do. We make this cap. We make a ultrasound system that could potentially be by the side of the patient. So we could go into an infusion treatment center. We could go to a very small private clinic where the space is very constrained, and we can open the blood-brain barrier, given that we have the MRI and CT of the patient. So we can get that from a previous, and we can build a patient-specific model of the patient, and we can deposit the ultrasound very accurately and open the blood-brain barrier just at that location. And this location could be as small as 0.5 centimeters by 0.5 centimeters by 0.5 centimeters, or it could be as large as a hemisphere or half hemisphere things like that. So we have very good control on the size of the opening. We have very good control on where it is being opened. We can go all the way down from the base of the skull all the way down to the brainstem for about 10 to 12 centimeters. So how does all this work? And this is something that was not invented by me, but invented by a group out of Toronto. So the way this works is that the patient is injected with these ultrasound contrast agents, which are commercially available. These contrast agents are typically used in diagnostic examinations, and these contrast agents are really comes in a little vial, and you inject saline into it, and you pull out the saline, and you inject that into the patient, as it is shown here in the picture here. These contrast agents are little micro bubbles. So the micro bubbles are 1 micron in size, very, very tiny. The vasculature in the brain is about 4 microns. There's no blockage of the artery of the vascular system or anything like that. However, when these microbubbles are within the system here, when the ultrasound falls in the brain, the ultrasound forces these bubbles to actually vibrate both in size and position. And the reason that it can vibrate in size is because the ultrasound is, after all, a mechanical pressure. It changes the ambient pressure of the tissue it is propagating through. And what that means is that if you can think of a balloon, and can blow into the balloon, and the balloon becomes bigger, that's exactly what's happening to these microbubbles. These microbubbles are vibrating about 1,000 times a second inside those sphere, and when those bubbles become very large, they actually force those tight junction proteins between the endothelial cells to break open temporarily for 90 minutes. So it's a mechanical disassociation of those proteins for about 90 minutes, and then these microbubbles themselves disappear in about 5 minutes. That's all, and it's gone. You only need at any location, and then the blood-brain barrier remains open for about 90 minutes, and then it closes back completely within an hour. Within 24 hours. So we can open the blood-brain barrier safely. Why do I say safely? These openings are just small enough to let large molecule drugs go through. So you can let 150 kilodaltons go through. You can get 200 kilodalton drugs go through. So previously, things like BCNU, things like cardioplatin, things like temozolomide, even those are 90 kilodaltons and above. Those are very hard to penetrate through the blood-brain barrier. But with this opening now, you can open that very accurately and let larger molecules like any of the and it's not large. Those openings are not large enough to let these blood cells go through, and that is important. So there's no bleeding per se. There's no microhemorrhaging per se. So the red blood cells don't go through, and and then cells from the tissue doesn't come into the blood, and that's kind of important in the context of thinking about the risk of further downstream metastatic migration. So coming back to the system here, it's a cap. It's a device that can sit, that's portable, that's quite inexpensive. It's about 100K, 150K to produce, and then we can open the blood-brain barrier. We can deploy it in their heads. People, I'm sure you guys know about Optune. You have to shave their heads. This is not like that. You don't have to shave their head, the patient's head, and the patient experience is 30 minutes or less. We have a great IP portfolio. I can go into any of the details later on, but our portfolio is all about safety, all about how to let the patient sit on a chair like this. There's no constraints on the patient. We don't we don't screw the cap to the patient or anything like that. The patient can move around. So that's the basics of what we are doing here. And then what are we going to do with it? So the go-to-market today is that when we open the blood-brain barrier, two things can happen. We can let the biomarkers come into the blood, and those biomarkers, and we can draw blood peripherally. So we can potentially open the blood-brain barrier non-invasively to let these biomarkers come through, and then we can sample the peripheral blood, in the arm, and then look at those biomarkers and understand the disease process in the brain. So that is what we are going after as our first indication. So there's no drug involved. We're going to be opening the blood-brain barrier to understand, the disease process, to know about the spatial heterogeneity, the temporal heterogeneity, look at disease recurrence, to rule out, radiation necrosis versus real progression. We can rule out those things, based on what the biomarkers we see when we open the blood-brain barrier like this. And therein lies the blood-brain barrier safely for the biomarkers. On the flip side, when it comes to therapy delivery, we can open the blood-brain barrier to let the drugs go out in the same location. I neglected to mention that current data is showing that we can open the blood-brain barrier safely without any adverse events, and I can come back and talk about how that's done here. But that therein lies the concept of precision medicine. We can open the blood-brain barrier to understand the disease. We can make therapy decisions. We can guide therapy decisions and disease monitoring. And we're going to start our go-to-market again is with what we're calling sono biopsy, which is non-invasive. We could biopsy of the brain and share very briefly. But some data here, animal data demonstrating that we can open the blood-brain barrier safely. Here's with our device that we have developed here. Here's a picture of a dog before sonication procedure. This dog has gadolinium, which is a proxy for a drug. Gadolinium, of course, does not cross into the tissue after we have reached the blood-brain barrier with our device. It's very punctate. It's a very small, accurate opening through the skull in non-invasive, and the dog was completely fine. This particular dog was sacrificed. So you can see Evans blue here. Evans blue also is a proxy for a drug. It doesn't leak out unless there's a breach in the blood-brain barrier. You can see that Evans blue leaking out at the location of the MRI, giving us further evidence that the blood-brain barrier was breached. And also, further H&E showed no microhemorrhaging, which again demonstrating that there was no leakage, there was no bleeding, when we opened the blood-brain barrier. So that was for the case of glioblastoma, but you also know that in the case of here's a picture showing that we can open the blood-brain barrier in multiple locations with our same device. So with the diffuse disease, which really, glioblastoma is also a diffuse disease, we can open the blood-brain barrier in a large region. And here we talk about accuracy, which we can. This is data with our device demonstrating that. So this was we radio-labeled Herceptin and Keytruda. Again, 150 kilodaltons doesn't cross the blood-brain barrier easily. So we took this. This was a hard study to do, but yet we got some pretty good results. With a non-sonicated site, you can see the uptake of the with a sonication and giving the same drug, we saw a twofold increase in the uptake of these radio-labeled drugs. I suspect that the uptake was much larger. The only reason we got a 2x increase is because there was a lot of time difference between when we opened the blood-brain barrier and when we took the animal down for the radio-labeled injection. But the H&E shows that there's not any extravasation of blood. So these kinds of studies we can do, and we have proof now that we can take large drugs through the blood-brain barrier. And now I'm going to show you some human data in the context of the liquid biopsy application. So here's this data was obtained by it is that they've taken 10 patients now, opened the blood-brain barrier just before the surgery, and the blood-brain barrier was opened again with a device that has a similar functionality to the device that we are building here. And these are the first 3 patients of glioblastoma. Here's a glioblastoma opening the blood-brain barrier. This demonstrates an increase in third mutation peripheral seizure. So what this is saying to us is that look, we can open the blood-brain barrier through the intact skull, look at the peripheral mutations that can tell us about the mutations that are present in the brain, and that can actually increase. And the reason this is important is we can now look at commercially available liquid biopsy assays like from Cordance One and Exact Sciences, which have a limit of detection, which is about here. So today, if the blood-brain barrier was not open, the mutation, the concentration of mutations in the blood, peripheral blood, is below the limit of detection, and thereby those assays cannot be used to understand what's happening in the brain. But with the blood-brain barrier opening like ours, those commercially available assays can now be used for understanding what's happening in the brain, because the concentration of those biomarkers can actually increase significantly, and thereby these commercially available assays can be used for diagnosis, which is exactly our first indication as given to us by the FDA, stating that indeed we can open the blood-brain barrier and look at these biomarkers. And when we're talking about biomarkers in neuro-oncology, we're not talking not just about the cfDNA, but also obviously the proteins come out, the EVs come out, and those have just so much information. And this is what we're beginning to understand, that with these, we have to build upon this data, and understand what's going on in terms of therapy monitoring, in terms of disease progression, and everything else that we talked about early on. And I'm not going to talk about neuro-oncology, but there's a whole new story about neuro-oncology, about Alzheimer's, Parkinson's, all that we can talk, offline. So just want to spend a couple of minutes on coming back to glioblastoma. So I just talked about, our focus currently is that can open the blood-brain barrier for glioblastoma. What we can do with it is that we can understand, we can monitor the disease, we can understand if it's recurrence or not, we can monitor progression versus pseudo progression, we can understand we can do the molecular characterization of the tumor. Again, this is all the things we can do with that disease currently. And if we know all this, then when we can understand when a drug comes along, and we can understand if the drug is effective or not. So this is our key opportunity, at least in the beginning, to get through the FDA to get out and do the diagnosis on a GBM patient and on a brain metastatic cancer patient. Now there are other devices that use ultrasound to open the blood-brain barrier, but those are all invasive and very difficult to use. But they are establishing some really good data. The mechanism of action is identical. So those data are relevant to us in the abstract here. But let me just kind of bring this group up to speed on some really exciting things that are happening in the area of focus ultrasound and glioblastoma. There are really two sort of big things here. One of them is that clearly we can disrupt the BBB the blood-brain barrier opening for the purposes of monitoring and drug delivery. We just talked about that. Sonodynamic therapy. Here, the blood-brain barrier is not disrupted, but the 5-ALA drug, preferentially accumulates within these tumors, and ultrasound can actually activate that 5-ALA and can cause these reactive oxygen species to actually accumulate within the tumors. So that's fantastic. Now there are about I think there are about 12 or about 20 human patients that have gone through this procedure, and the results so far are really pretty phenomenal. And it's not just about GBM. Some of the patients have had DIPG, but really half of that data set is GBM patients. 5-ALA drug, and we'll be starting some clinical studies on this in about a year's time. These are very exciting studies that are going on, and potentially this can be very useful for the patients here. So there are two companies that are out there that are doing these studies, Cartera and Alpheus, and Cartera is using the blood-brain barrier disruption technique. They are using carboplatin, to deposit carboplatin, and they're getting very high levels of carboplatin within the tumor. It's an invasive device, and the carboplatin, and they're seeing a modest improvement in the median survival. I suspect the modestness is because of the fact that the Cartera is a very is a unique device. It's very focal in nature, and we know that GBM spreads rapidly, and I suspect that's what's going on. But with our device, we can open the blood-brain barrier, in the whole brain setting. It's using 5-ALA. They just started their studies, and they have a really nice abstract demonstrating that in the 5 or 6 patients they've done, they've seen significant improvements in the survival rates. So very good improvements, and with our device, we can enhance the effectiveness of these two therapies. Okay, so back to our device here. I talked about what we are doing. Our first application is in diagnostics, to open the blood-brain barrier to carcinoma, but also for brain metastatic cancer. Here's our pipeline. We are working on brain tumors, as I just said, working with several partners. You'll recognize many of them here. We've also begun a therapeutics program with multiple partners, and in the next 1 year or so, you'll be hearing a lot more about these efforts here. Just to put a fine point on our business case, if the patient were to have an overnight stay and significant costs, that's the picture in gray here, and then yes, it can be done. It has not a non-zero risk of infection and potentially death. Whereas in our case, if a biopsy is needed, you can go to a clinic, put on the cap, a 30 to 60-minute procedure. You're done. You draw the peripheral blood, and the patient goes home. So significant, economic, advantage that can be obtained by our device. I know I'm running short of time, but this ultrasound, we've got other devices, but these are all, like I said, either very invasive. Insight Tech needs an MRI to guide ultrasound. Every time the patient has to be within the MRI system for 4 hours with the helmet screwed onto the patient's head. Really difficult for the patient and the institution. Cartera, I talked about this, is a craniotomy device. It needs craniotomy, and it's very focal and doesn't address whole brain diseases. And then Nanophus is a very large robotics-based system, and it's really not conducive to large-scale optimization. So with these 3 leading companies, I would say, opening up the field for us, here we are. We have great way of using that science and delivering ultrasound in a non-invasive manner. That's what we're doing. Let's keep over the side. As you all know, the SAM and TAM are large. No need to go through this. I'll just mention that we have some disposables that make up our revenue. Again, we can go through all this at a later time. So let's see. So one of the things that I didn't mention is that, for glioblastoma, we can potentially take existing drugs that have failed, phase 3 because they haven't crossed the blood-brain barrier, and we can repurpose those drugs, and that's a huge potential. Bryan talked about the tumor microenvironment. Yes, we can actually look at that on the aspect of with these drugs and what happens at tumor microenvironment. So we are raising funds. I'm happy to say that we are oversubscribed, which is great. We got the money right now is to convert our device that is working on animals to make that safe to be put on a human, and the goal is to get ID submission by the summer of next year and get into human trials by the end of next year, beginning of 2027. I'm also very happy to say that NIH has for the human study, for the sonobiopsy study. So we got the $8 million covered, and then we got there's $2 million, and we are slightly oversubscribed. So not almost at the end of our current fundraising, but we will be raising towards the middle of next year for this larger pivotal study, which will be 85 patients. So please keep us in mind when it comes to that. We have a great patent portfolio all about how to use a system safely and non-invasively. And I would be remiss if I didn't mention my team, just a fantastic team. You're all welcome to come and visit us. We are near Stanford, 5 minutes from Stanford. Happy to give you and just in summary, we're raising $8 million, and we're almost done raising this money. So that's what I have to say today.
Excellent. Thank you. You're actually quite early.
Oh.
So we have about, thank you for giving us some time back. How about we spend about 5 minutes if anybody either online or here have any question? Yeah, Tracy.
Wonderful presentation. I just had a very quick question. I saw that you can use the ultrasound in order to diagnose glioblastoma versus using a surgical procedure. Are you able to determine whether the tumor is methylated or unmethylated through this process?
Yes, we can do that too. Yeah, I know. Yeah, I didn't talk about that, but yes, I know that's the possibility is really great. And it's, I mean, we can go into that in detail, but definitely, yes.
Yeah, I'd love to connect with you later because we're treating glioblastoma right now.
Okay, will do.
Excellent, thank you. Anyone else? Yeah.
Who are some of your current investors on the cap table?
Say again, please. I couldn't hear you. Say again.
Who are some of the investors you have on the cap table currently?
Oh, investors. So the lead VC is Sonder Capital. They are putting $4 million. And then on top of that, there's Acorn Bio, who is putting another $1 million. And then the Sontag Foundation is coming in with some money. And then the NBTS is also coming in with some money. So the foundations and some large VCs are coming in.
Thank you.
Great.
Yes, please.
Oh, we got another question back there.
And there might be one online. But,
okay.
One question regarding dog patient studies. You did study in healthy dogs and pigs. Dogs have glioblastoma as well, and as far as I know from my boss, she's an MD PhD as a CRO in Hungary. They are the glioblastoma of dogs is more similar to child's glioblastoma.
Yes.
And hence, I was wondering whether you are planning to do one of your in vivo diagnostics and maybe therapeutic studies in dog patients.
Yeah, I would love to. It's actually quite difficult to get that model in the US. There are only a very few centers that have dog patients, and you have to have a pet with the glioblastoma to actually there's something. It's very, very difficult. So if you have a source where we can do these studies, I would just love to connect with you, because you're exactly right. That can actually tell us a lot about human glioblastoma, and for the dog itself, it actually could be very useful there as well.
Mm-hmm. There is an existing vet network because you need the vet to
Yes.
recruit these patients. Huh?
Yes, yes, yes.
Just to piggyback off of that, UC Davis Veterinary School is looking for something to help their dogs with glioblastoma.
Yeah, I've talked to UC Davis. It's you have to have a sponsor within UC Davis for this to happen. So, and I haven't been able to get a sponsor within UC Davis. But if you have somebody I can talk to, I would love to talk to them.
I can introduce you next week.
Thank you.
Excellent. Thank you. Great. Any other questions? Was there a question online? Okay. If not, we'll take a quick 10-minute break, and then this will give us. So right now it's 3:26. We'll come back in 3.
Ticker symbol is MAJI. Website's there. Next slide. Forward-looking statements. I got to give this, so we'll just read that at your leisure. Now, I'm a military guy, Air Force Academy guy, so I throw in military analogies. That there is the enemy. That is a glioblastoma cancer stem cell. And what we've been doing for decades, trying to defeat that enemy, and it's elusive. It hides from us. It sneaks around. It evades. Does all this stuff. So I am just, and that represents. We found out that represents the last cell. How many people here have heard stories of glioblastoma patients where it's gone, and then it comes right back up. See, there's a 90% recurrence with glioblastoma, and why? That guy. So what gives me hope, where we're feeling lots of despair, is that I think we finally have a solution to target that guy and give people, cancer patients, the quality of life that they deserve. That's my little preamble. So now we get to the good stuff for the investors. I'm going to read this a little bit, but we've patented exosome technology. It delivers genetic therapies where many drugs can't go, and we've heard it in these other presentations about the blood-brain barrier. We showed that it can turn drug-resistant brain cancer into treatable disease. $1 billion potential, talking oncology, neurology, and diagnostic. Our patented manufacturing process creates high-quality exosomes at scale. That's been something that's been bugging my exosome industry for a very long time, solving production challenge that's basically stumped others. And here's the cool thing. We let the cells do the work for us. We keep it simple, and we're poised for blockbuster success, we think, here. And then we have a clear pathway for orphan drug designation, and we are ready to scale. Now, the top challenge, I said challenge, but it's challenges, is most people think that drugs are unable to cross the blood-brain barrier, and that's the main issue with glioblastoma. But it's really what we heard in the first slide presentation is our ability to kill the cancer stem cells, okay? But it's really both, and our solution looks at both. And the exosome market is $3 billion. These are some, nice footnotes, if you would. A competitive advantage is our patented loading tech, blood barrier, brain barrier penetration, and permeation. You'll see that. We've got some nice proof of that. What are we looking for? This ask is so astronomically low. Look at that, $2.5 million for an IND. And then how are we going to exit? We could acquisition, spin-off, licensing deal, phase 2A. So now I set the stage, so we're going to look at what the problem is. The problem is the inability to destroy cancer stem cells. And you'll see there's two graphics there, chemo, the radiation. And what that does is it shrinks it, but the cancer stem cells survive. And then what happens? It comes back again. And then if we target the glioblastoma stem cells, what do we see here in this graphic? No relapse. And that's the idea, is we want to get rid of this recurrence. And you'll see the third line, you know, TMZ works, goes through the blood-brain barrier. Problem is, it's not getting to the root of the problem, which is the resistance in the glioblastoma stem cells. And you're going to see that I think we've solved this problem. Next slide, please. Now, new therapies can't get into the brain, and that's why we have, you know, the technologies presenting here, you know, because they're trying to, you can see the graphic. We're trying to solve that. I put a survival curve in there. Man, look at it. It's like 5 years survival, 6.99%. You've seen all the other things. This is a big problem. But new therapies can't get in the brain. Our therapy can, and that's what you're going to see. And you'll see my footnote at the bottom, billions wasted on ineffective therapies. Next slide, please. So now that we've identified what the problems are in glioblastoma, let's take a look at what some of the potential solutions are. If you look at this slide, you're saying, oh, wait a second. I don't know if you can get rid of that thing on the right, but no, it's fine. We basically know how to solve this problem, okay? We know how to kill these glioblastoma stem cells. The challenge that we have here is basically delivering that genetic payload and doing it from the peripherally in a minimal invasive way, not like sticking probes in people and, you know, intratumoral injections and all that sort of stuff. So you see there's different approaches. The last approach there is NINOG, and you see, I like to highlight, NDA required for preliminary results, extremely encouraging. We do have preliminary results, so if any investors out there, let's get the NDA signed and let's see some amazing stuff that we have behind the curtain, so to speak. And what we have, the other things we have out there, nanoparticles are degraded by the liver, intratumoral injections, like I said, was impractical, and the viral vectors, you know, they're difficult crossing the blood-brain barrier. And with exosomes, they all make it through? No. Do a lot of them make it through? Yes. And that's the difference. When I talk to my scientists, when we look at like the viral vectors, we're talking really small percentages. We're talking like orders of magnitude different when it comes to exosomes. Slide, please.
Not working now. It may.
Oops. Is that good?
Okay, now it's working. Thank you.
All right. So remember, I talked about, you know, the TMZ resistance. Now we're going to talk about the blood-brain barrier. We've engineered exosomes, which is basically the body's natural delivery vehicle to carry large genetic payloads safely across the blood-brain barrier and target cells directly. So it crosses it. We can have these large payloads and deliver complex molecules. It's precision. We go right to the right tissues that we want to. We could train these exosomes to do that, low immunogenicity, and then scalable manufacturing. So in vitro, we showed that we had 3 times the GPM efficacy boost, and I'm going to show you that slide shortly. We have an efficient brain penetration, and we've proven it, and you'll see that in some of the slides I'm going to show you. And then orphan drug application is filed, so we're very hopeful that we're going to hear some news on that very shortly. And you'll see on the right, I have drug delivery platforms. So what do? We load, we target, we deliver across the blood-brain barrier. And let's go on to the next slide. So some people in this room may or may not know what exosomes are. So exosomes, they're just like little pockets or sacs of basically cytoplasm from that particular cell. They're 30 to 100 nanometers in size and carry proteins, lipids, genetic material from one cell to the other. So you can kind of see they're like encapsulated into themselves, and then they make their way into the world. To give perspective, an exosome would be a speck of dust on that basketball. Next slide. Go through these kind of quick. A lot of this is I just repeated. We can, the third one down, stem cells are used, these acts as armored car couriers. Think about having very, very high value cargo in an armored car, and it's going from one location to the other. You can think of exosomes like that. And let's go to the next slide. Are they important? This is a little bit of a repeat as well. They act as messengers. They affect cell function, behavior, intracellular. So they do really localized stuff, but they also do long distance communication. They're crucial in healing. One of the things that I'm just kind of throwing in there is we also have a commercial side of our company, and we use mushroom exosomes. They contain a lot of vitamin D, lots of great healing properties for like the skin. And they play major roles in diseases. I don't know if you've heard tumor-derived exosomes, but that is a thing, and that helps increase metastasis. And it can be used as a diagnostic, so we also have a diagnostic component to our company, and a treatment, kind of like monoclonal antibodies. It's just kind of a gee whiz. Did you know that all plants and animal cells, all, can't say that in science very often, but all have exosomes. And they first came about, people thought that they were just garbage sacks of nuclear material, but we found out it's much more. Let's go to the next slide. This is kind of a graphic of what it is. You see there's no nucleus in an exosome. It's just nuclear material. That's it. Lipid bilayer, but it's different than extracellular vesicles, different than nanoparticles, because it contains the outer surface of an actual cell membrane. Slide, please. Okay, so we're going to get into the good stuff, right? Everyone wants to, yeah, I saw a thumbs up there. That's right. We're going to get into the efficacy. Let's go to the next slide. In order to do that, I want to make sure we're all on the same page and we understand what we're talking about here. So glioblastoma stem cell prevalence. It was a study. You can search it at the sources there. But what it did is it showed you that there's 4% cancer stem cells. And you're going to see the significance of why I'm talking about 4%. So let's go to the next, I don't know if you can see, so let's go to the next slide. This talks about the TMZ mechanism. I think we kind of already beat this through a pulp. The TMZ comes in, it wants to destroy the DNA, and then the problem is that the MGMT, fixes it in these. The MGMT mechanism helps fix it in these cancer stem cells. And then, there's the methylated on the left-hand side and the unmethylated on the right. And if you get unmethylated, you have a poor response. Now look at those numbers there: 43% unmethylated, and then 24.5% it was unknown. And there's a study over there. Man, just quick math, that's like over 60%, right? So over 60% of the people with GBM are encountering this type of resistance. So this is the root heart of the problem. All right, let's go to the next slide. My scientist, if he's out there listening, this is for you. These are in vitro results. Okay, he wanted me to emphasize that. But what I will tell you is what else and I'll explain this chart in a second here. This is only glioblastoma cancer stem cells. We're not looking at cancer cells or glioblastoma cancer cells and cancer stem cells. We're just looking at just that one subset. Okay, so the black represents without our shRNA. You'll see that. So that's basically we're just doing chemo. Okay, so black represents chemo, and the amounts of chemo on the bottom are increasing. And then the exosomes is basically exosomes plus chemo, and the chemo is TMZ, right? And man, man, this is just so cool here. I mean, look, so in vitro results demonstrate a combination therapy. There was a silencing gene, so that's what we deliver, a silencing gene that reduces the drug resistance mechanism of GBM and help kill almost 100% of the cancer stem cells. Like almost. You can kind of see the top band of that far one. So we're using a normal level of GBM treatment, or I'm sorry, TMZ treatment, and look at what normal is without the exosomes. It's a black line there, 25%, which that's with most of the studies out there, around 25%. And look at that. At 1/100th the dose, that's the first arrow. Look at that killing capacity. That's 3 times what TMZ can do. So 1/100th the killing capacity, and with exosomes. So we really are working on that resistance mechanism. And then here's what's so exciting. It was non-significant. But look at 0 chemo. Look at that blip up of red. Those were exosomes. So there was a little bit of killing going on there. And I attribute it to just common sense. Is like, well, what happens if you take away the repair mechanism? Well, don't cells normally die on their own? Yeah, if you make them normal. So maybe that's what happened there, and that's what explains that. Go to the next slide, please. So I got the 4 Ps. Every good biotech guy's got to have this analogy. So here it is. We penetrate the blood-brain barrier. We permeate into the brain tissue. We paralyze, and I'm lying there a little bit. So we block the repair and proliferation mechanism. And finally, we prepare, which is basically sensitize the brain for the TMZ onslaught. Go to the next slide. And so we're going to take take these now in that order. But first, I got to tell you like what it is. So what are we doing? We are giving gene therapy to cancer cells. So this is our breakthrough technology. There's our patent, and we use mesenchymal stem cells to make our exosomes. You'll see the plasmid is entered in there, and now we have a highly decorated, targeted exosome with the nuclear payload with the shRNA in it. Next slide, please. Patented loading process, and this is a little bit of the same. So receptor targeting, genetic payload, and then remember what I said. Hopefully, remember what I said in the beginning. The cell does the manufacturing for us. So that's what's so nice. So when the FDA is looking at it, well, who's making it? The cell is making it. There's no impurities in there. There's a whole lot less steps than what maybe some of our competitors have to do. Go to the next slide. Okay. This is another thing that I need to. This was done in mice, and we used green fluorescent protein, and we delivered it with a subcutaneous injection to the mouse. And what you'll see is it penetrated the hippocampus region of the brain, also the cortex of the brain. So what's cool there is the bottom line is, subcutaneous injection's cool, but man, what about an inhaler? It opens up that possibility. We could go that method of distribution as well. Let's go to the next slide. Our drugs are targeted with a brain homing peptide. We tested BHP and MCAM, and what you can see there in the left is you have an empty exosome, and obviously you're not going to see any fluorescence. B, you just have an exosome with the fluorescent protein in it, and you don't really see it really light up. But now, put on the peptide and look at how that targets it and makes it active inside the cell. And NCAM does the same. So you can see some of our fluorescent studies here. So let's go to the next slide. This is an organoid, and some of you that went on this little tour with me saw organoids. This was our organoid, and we just show you the staining so you can see it's consistent through and through. I mean, that's the whole purpose of the top half of the slide. But the bottom half of the slide really tells a story, okay? So if we take our exosome with no decoration and put a red fluorescent protein in it and deliver it, look what happens. Nothing. You see, without the targeting, the permeation is highly localized. Look what amazing things happen when we take the brain homing peptide and decorate it on the exosome. Look at that. Look at that permeation. You can kind of see how it makes its way through the entire brain tissue there. Let's go to the next slide. Improved exosome delivery was functional when it hit the brain. You'll see here, the black void on the left. And this was, by the way, an in vitro test. There's nothing there, and until we actually put the homing peptide on there, you can see now it became functional. So go to the next slide. These are typical GBM or mouse models. And the reason I'm going over what everyone else is doing, so you can see how rigorous our preclinical trial data, which I'm not announcing here, but we can talk about via NDA. So xenograft models, implantation of GBM in immunocompromised mice. That's what we did. But these are all the other ways that people study GBM right now. And the most favorite way is the GEM model down there at the bottom, where these mice spontaneously get GBM. And then what we're doing is we're really trying to cure mouse GBM. We're not really curing human GBM. So we wanted to go a step above, and let's go to the next slide. And this is kind of a preclinical trial design that we had. So we're using humanized mice implanted with cancer stem cells. We're doing a couple groups, and the groups we're doing is control, the GBM, and the exosome. So very similar to what I just showed you on the in vitro data. So we're just basically doing that, but in mice. So the reason I show you this slide is so that you know when we get data. I hate to say this, but the barrier on this preclinical trial was so high, it's almost like it's doomed for failure. But what if we succeeded? Then we know we have a really robust model. So hopefully, you'll take a walk on the wild side with me and the investors out there, and we'll sign an NDA. Next slide. So druggable target. So now I want to show you a little bit about our druggable target, what it is, and it's been mentioned in one of the other presentations, but we'll just look at it from a big picture. Next slide. What is NANOG? It's an embryonic gene together with OCT4 and SOX2. On the left, you'll see stemness qualities are right there, and you'll see there's like a little triangle, OCT4, SOX2, NANOG, and those genes create the stemness in the glioblastoma stem cells. And then NANOG, what's it responsible for? The cell renewal, proliferation, metastasis, and chemical resistance. Correct. Now, just from a big picture, embryonic stage of life is 10 weeks or less, right? So right now, all of us, hopefully, guarantee none of us have NANOG in us on a detectable scale, okay? Because if we do, we probably have some problems. All right, let's go to the next slide. NANOG expression in cancer stem cells. You'll see the second bullet point there, pseudosine variant. That's what we're actually, that is precisely what we're targeting with our RNA. And go to the next slide. Call this a master regulator of cancer stem cells. there's a tutorial of NANOG. I think it's down at this green thing down at the bottom. And it's the master regulator of pluripotency. It's abnormally activating cancer stem cells. And you'll see the chart there. NANOG drives the cancer stem cells, all the bad things we talked about. And then in the far lower right, you'll see NANOG gene expression by tumor type. Look at that. We haven't even touched osteosarcoma, but pancreatic cancer, colon cancer, breast cancer, glioblastoma. Glioblastoma is just low-hanging fruit because our exosomes can make it through the blood-brain barrier, and all that. But there's room to grow just by looking at this because the gene expression is in, how do I say, the nastiest of cancers out there. The nastier the cancer, the more the NANOG gene expression, right? So we are a company that's essentially going after the nastiest of cancers. Bring it on. Next slide, please. I gave you a corporate overview for the investors out there so you know that right now we're just talking about the clinical stage biotech, the middle, the guts of it. But we're commercializing applications of plant-based exosomes while advancing the diagnostic and therapeutic potential, right? So just know that we're more than just a one-trick pony on biotech. We also have commercial aspirations. We also have diagnostic aspirations. And we have some very, very good technology. You sign that NDA, and we'll probably be talking to our scientists. Next. Clinical stage biotech. Our pipeline, it's really kind of not developed, but it is what it is. We're starting off here, but you'll see on the bottom there is topical dermatological prep. So we want to get this. We want to get our exosomes approved as a device. Exosomes along with a device, and that's also a pathway to approval. Next slide, please. Orphan drug application. I mentioned this that we filed it, but I want to bring this point out. The bottom line is, look at that. Typically, these fetch over $100 million valuations. So, what are we doing at a $5 million market cap? We're waiting for someone to give us that designation. And once we get that designation, feel very comfortable in, we would expect to see a value inflection play and a major one when you look at what's typically in the private market they garner. Next slide, please. So our glioblastoma model timeline, preclinical study, that's kind of where we're at now, and the CGMP, we need to do that. We did file our orphan drug application. We're hoping to get the acceptance of it. And then look in the lower right, one thing that I want everyone in this room to know, that we are open for business in terms of compassionate use. So we are completely open to doing compassionate use. You come, bring a glioblastoma patient to us, and we will help the investigator get that IND and move things forward. And we think that we think this is the right way to go and most efficient way to market. Next slide, please. So ground floor opportunity, a new class of therapeutics, that just boggles my mind here when you look at the statistic. I'm also a little bit of a biotech research analyst, and I did a little bit of research. The phase 1 and to phase 3 trials of all drugs out there, around 9,000. How many, I was curious, how many of them are treating exosomes? 10, okay? So right now the market, in terms of market share, is segmented between small molecules and monoclonal antibodies. So monoclonal antibodies overtaking small molecules. I think it's 45-55 split, somewhere around there. But I think there's room, if you would, for a third party, okay? And this is, and I think this could be it. Okay, I think this could be it. Now look at some of the cool things that we can do here. The government is supporting exosome research. The pandemic created all these vaccine diagnostics, and a lot of research came out of the exosomal research. A lot of new developments, including some of ours. Look at the upper right. New manufacturing advances like freeze-drying exosomes. Exosomes aren't alive. They can be freeze-dried. And payload large scale. And we can have our mesenchymal stem cells make these exosomes. And that's some proprietary technology. And then we have some diagnostic markers to detect cancer really early. What if I told you we think we could detect cancer at the 1 millimeter stage? Wouldn't that be cool? I'm not saying we are, but what if? I always do that. Next slide. Large market potential. Global nucleic acid market was valued at $41 billion. This is just large market. I mean, if we're in this room, we all know large market. Next slide. Again, the exosome market now is big too, and the exosome market is $3.2 billion. Next slide. So the future value drivers, I call them catalysts, orphan drug approval, out-licensing opportunities. Love to do a phase 1 trial. All this takes money, and then don't discount our ability to look at medical devices, get the exosomes approved as a medical device. Next slide. Got a lot of competition. NKore is probably, in my opinion, on the therapeutic side, most advanced. They have something for Duchenne's muscular dystrophy. And there was the FDA guy that manages that division. He didn't speak well about their exosome therapy, and all of a sudden, 4 weeks later, he's gone. So we got to think that people, the administration's friendly to us, our industry. And then another one down there is Neuro-oncology. They treat spinal cord injury. You just go onto their website and you look at a mouse that's completely, they break the spinal cord of the mouse, and then they can't move. And then the other one they treat with exosomes, and it's just walking. A video is 1,000 words, right? And then Athlon, what they do is they have a device that basically filters out exosomes from the blood. So it's you hook it up. It's like the excorporeal membrane, the heart lung machine where they circulate the blood and they take it out. So that's our competition. Nutshell. So next slide. And this is about the company. There's a 2-man team, reverse merger. There's the statistics on the share count. Got a very thin float. 6 million shares. Actually, that's wrong. It's 8. And then our market cap is actually a little bit less than that. It's a little over 5. Okay. And I think that is the end of my presentation.
Thank you. Actually,
how much time did you take?
You took 32 minutes, so not bad. Excellent. So we have room for perhaps a few questions.
And just one maybe, just in terms of your preclinical study, for the exosomes, are you using what's the contents of those exosomes, and then what are the?
Yeah, excellent question. We used cancer stem cells, and maybe I didn't drive that point home because I prefaced it with the 4%. We didn't use 4%. We used pure. We used concentrated stem cells. So that's why I didn't get into that. That's why I thought that when you look at this, that study's designed to fail. Wait a second. You're using pure cancer stem cells injected into mice, and you're trying to treat them. That's what we did.
I think Richard was asking about what was the content of
the exosomes themselves. So are they targeting?
The cargo was Sorry, I didn't. Thank you for catching me on that. The cargo was shRNA, and then it was nanog. It was, you saw on the slide, it was called nanog P8. That's the exact. Yep. Mm-hmm.
And then was it targeting, or was there targeting?
Yes, there's a targeting component. It's called the brain homing peptide.
Okay. So
the brain homing peptide. Do you remember the red slide where it permeated? So it would use the brain homing peptide because the idea is it's going to permeate and hit. Not only it's going to hit all the cells, all the brain tissue, or as much as it can, and that includes normal brain tissue as well as glial. Because remember what we're on the hunt for. We're on the hunt for that last cell. And the only way we can do that is to make sure we cover everything. Pardon the expression, we got to carpet bomb the brain and sensitize all the cells to, all the cancer stem cells to the TMZ that's ready to come in and mop everything up.
When you be sensitizing the whole brain in that case as well?
The whole brain doesn't contain nanog. Only the cancer stem cells contain nanog.
Any stem cells.
Only the cancer stem cells contain the nanog gene. Yes. Cancer.
Like glioblastoma.
Right. Other stem cells don't possess the nanog gene. Remember, the nanog gene is like an embryonic stage quality, right? It shouldn't be in our body right now. Okay? It is in very small amounts. I talked to our scientist, and he said, Mike, what happens when you break a bone? Okay, nanog comes out to play. It does. But if you can detect it, and that's the beauty of our cancer detection thing, and that's another element of our company is the cancer detection. I got a whole new slide deck on that, but that wasn't the purpose of this. Is we can detect it, and we feel we can detect it very early. Because if you have nanog, you have something in your body that shouldn't be there. Good question. Go ahead.
One question. Can you tell us a little bit more about manufacturing? Because it's not so simple with exosomes and then loading it. Where do you do it, and what is the...
That's what that NDA note was about. So yes, I'd love to tell you, but it is very much something that defines us as a company. So yeah, it's.
And that's why the market is still so small.
Yeah, that's why
a lot of Europeans were a little bit ahead of us.
Yeah.
And we'll see a few where there are a lot of exosome societies, as you know, in Europe.
November, I think they asked me to talk at one of them, in what was it, Netherlands?
Either the international or the new.
Yeah. Okay. Wonderful.
Fantastic. Okay. Well, we have our last speaker, certainly not least, and Tracy will tell us about her path, and as you probably can see that we have 4 companies at 4 different stages at various part of their journey, and it gives you a good overview, and then also the entire vertical from what you do with preclinical studies to possible delivery, diagnostics, therapeutic space. A lot of excitement. So I'm looking forward to listen. Oh, there's somebody. Is there a question online, by the way? I bring this up.
I think we're good. It's good.
The down's not working. Okay, it works. Hello, everyone. My name is Tracy Ryan. I am the co-founder and Chief Communications Officer of NKore Biotherapeutics. But to understand how I got here, I need to tell you where my journey began. And that was 12 and a half years ago when my daughter, Sophie, was diagnosed with a low-grade brain tumor at 8.5 months old called an optic pathway glioma. These tumors are traditionally survivable, but they have an 85% recurrence rate. So Sophie had a very, very long journey, one that she is still going through today. Sadly, when Sophie was first diagnosed, I immediately started diving headfirst into as much research as I could about these tumors, trying to find out anything and everything that there was to know about them. And one of the most defeating things that I found out was that only 3.8% of all government funding at the time was going to pediatric cancer research. And virtually none of this was going towards research for tumors like my daughter's. The drugs that she was put on were 40 years old and had been changed very little in how they were being used, which was, again, why there were no advancements and no real therapies to treat these children. When it all started to shift and change was in 2018. This was 5 years after Sophie had been diagnosed. At this time, I was the CEO of a company I had started called CanaKids. Prior to that, I was the CEO of a media agency. I'm a marketing executive by trade, graphic designer for over 31 years. I build other people's brands, essentially, is what I've been doing for most of my life. When Sophie was diagnosed, I moved into medical cannabis after film star and former talk show host Ricky Lake had begun filming our family on our journey. They filmed us for 6 years for a documentary that was on Netflix for 5 years, and it was about using medical cannabis in pediatric patients. We became one of the more well-known families around the world for the use of this plant that was guided by nurses and doctors and scientists that helped me develop a line of products called CanaKids. We were specifically looking at children with cancer, autism, and epilepsy, and we became known for some of the most incredible success stories around the world, especially in cancer. We were treating many, many cancer patients. Some people would come from all over the world just to get the oils in California and work with our practitioners that would guide this care. And we had patients that had been sent home with no chance of survival that using cannabinoids alone became cancer-free. But one of the things that we found was that when patients would stop taking the cannabis, the cancer would come back. So I knew that not only was I onto something, but there was something else that needed to be figured out in order to keep cancer at bay long term. So I really dove headfirst into science. And when, specifically in 2018, when Sophie needed her third brain surgery at the time, I was able to procure her live brain tissue from Children's Hospital Los Angeles. I was actually the first parent in the history of the hospital to walk out of that hospital with live tissue. And I fought them very strongly, went to the CEO, who then went to legal, who then went to pathology, and now they do anything I ask them to at that hospital. I have been known to use the media to my betterment, and has been one of the things I'm known for is my Rolodex and getting stories in the media. I am a fighter. You don't tell me no when it comes to my child, and I will stop at nothing to save my baby, which is why I am where I am today. It was because of this incredible woman, Dr. Anahi Jewett, who we sadly lost about 6 weeks ago to a tragic death. She is one of the world's leaders in the field of natural killer cells. She was in this field for over 35 years out of UCLA. Many of the terms that are used in natural killer cell science today were coined by Dr. Jewett. And it was when I was looking for a scientist to put these live tissue cells, these the tumor cells that I've gotten from CHLA into live mice, that I found Dr. Jewett. When I found her, I wasn't initially working with her. I was actually I'd raised several $1 million for the Technion Institute in Israel, where they could legally study cannabis in autism and in cancer, and was driving a lot of that research with the funding that I had brought into Israel. I was going to hand deliver these mice to Israel so they could do cannabinoid sensitivity on Sophie, so we could figure out better cannabinoid profiles to actually target and kill her disease. Because despite the fact she was one of the more well-known children around the world for the many, many, many successes that she had seen using cannabis, her tumor kept coming back, and we just didn't understand why. Whereas with many other neck-down cancers, we were seeing incredible, incredible successes, but not in my daughter's. And we were baffled as to why this was happening. Dr. Jewett saw the passion, saw the determination, saw the fact that I was not going to give up on my child, and she said, I'm going to help you. She said, but I have one caveat. I want to study your daughter's immune system. The things that you're telling me about medical cannabis are very fascinating. I've never looked at it, I've heard about it, but this has never been on my radar. So if you will let me study your daughter's immune system, I will give you humanized mice, not your typical immunocompromised mice. These were mice that pumped actual human blood. Their immune systems were almost identical to that of human beings. Compared to all the other mouse models in her 35 years of research that she had studied around the world, these were the best models she had ever seen and the most like human beings. So I thought I'd hit the jackpot. She called me one day and she said, Tracy, I don't understand what I am seeing in your daughter's immune system. I was like, what do you mean? I was bracing myself for impact because at this point, all I had known was getting knocked down over and over again and just trying to climb my way back up. But what she communicated to me is that Sophie's natural killer cells were that of a healthy person. And in the hundreds and hundreds and hundreds of patients that she'd studied over the years, she had never seen someone with cancer have as high functioning natural killer cells as my daughter did. So it made absolutely no sense. It wasn't until we were able to get fluid off of Sophie's brain that we discovered something really quite incredible. But before I tell you about that, I want to tell you a little bit more about what my daughter has gone through on this journey so you can start to get a sense of who I am as a mom, as a person in business, and as an advocate who is driven by helping human beings. My daughter failed 8 different protocols of chemotherapy and clinical trials over a period of 7 years. She's been fighting at this point for 12. That was the timer, I apologize. She's had 11 surgeries in total. I actually need to update this because a few weeks ago she had her 6th brain surgery for a VP shunt. It didn't have anything to do with her tumor. It was from the 5 previous brain surgeries that she had had where scar tissue had formed and was blocking the reabsorption of the CSF, the fluid our brain floats into the body, which caused hydrocephalus. It came out of nowhere. We didn't expect it. It was horrible, but so has most of our journey throughout the last 12 years. My daughter does have a left-hand side weakness. She is legally blind. She's had dozens upon dozens of hospital stays. She had a piece of brain removed to stop seizures that she was having, and she has now had over 75 MRIs. However, when we got this brain fluid that you see here in this picture, I got this CSF over to Dr. Jewett, and an absolute miracle happened. We figured out why Sophie had brain cancer. Despite the well-functioning natural killer cells she had from the neck down, she had no NK cells in her brain whatsoever. 0. Highly functioning CD8 positive killer T cells. All of her other immune cells in her brain were working beautifully, but there were no natural killer cells. And what Dr. Jewett discovered in her over 35 years of research is that it is natural killer cells that protect us from cancer. Natural killer cells can identify cancer stem cells. And when they start to act up in the body, the natural killer cells deploy and tell all the other immune cells what to do. They are the generals in the marching army. They are able to target and kill those cancer stem cells before they mutate into actual cancer. Once those NK cells have failed, and those failures can come from a myriad of things, obesity, poor diet, genetic mutations, environmental factors, horrible sleep patterns, extremely high stress. Stress is the silent killer. When those NK cells break, that is inevitably when cancer begins to form in the body and other immune-related diseases. This is what led us to forming NKore Biotherapeutics. The beauty of natural killer cells is that they have this extremely unique safety profile. They have the potential to kill both high-grade and low-grade tumors, solid and liquid cancers. Now, we have a lot of what we like to call junk companies out there today with natural killer cells that are using the wrong kinds of natural killer cells, or they're not activating them properly. And these cells do not have the cytotoxicity or the ability to stay alive long enough to actually attack and kill these cells, these cancer stem cells and these stem-like cells. That is not what we are seeing with our natural killer cells. And what's even more exciting is these patients who have had failed immune function, we're watching their immune systems be reestablished. I'm going to show you some data on that in a minute, but our patient that has gone the longest since treatment is patient 1 and patient 2, which is my daughter. In patient 1, He has been over a year without an NK cell infusion. My daughter is still getting active infusions. He's not had an infusion since July of last year. His NK cells are now holding steady at 21%, and his own body is killing the cancer right now, which is absolutely incredible. We've also published on the ability to accentuate other treatments like chemotherapies. We also believe that this is going to be a great add-on for radiation in glioblastoma, where we give some low-dose radiation at the beginning. We create antigen release, which is waving that white flag to the natural killer cells. The natural killer cells deploy, and then we see reversal of disease. We are not trying to claim that this is a silver bullet. We believe in combinational therapies. We believe that if we catch some diseases early, that this could be a monotherapy, but we are very excited about combinations with drugs like TMZ for glioblastoma. Here is some of the preclinical work that we've done in GBM. Now, we have an entire deck of our glioblastoma studies. This is a very, very small sampling of it since we have limited time today. This is using a supercharged natural killer cell treatment where we watched it decrease the number of glioblastoma stem cells and increase the number of dead cells in 3D tumor sphere models. The NK cells were added to the GSC tumor spheres with an NK to GSC ratio of 10 to 1, and these images were acquired using an inverted fluorescence microscope 4 hours later. You can see with the propylium iodide the penetration of the SNKs and the dead cells that are starting to come off of this GBM stem cell. These are some findings, some research findings that I've kind of summarized here when looking at the stem-like cells for glioblastoma. In the picture on the right, you can see that with primary natural killer cells, which is the center row, these are functioning primary natural killer cells. A primary natural killer cell is a natural killer cell from your own body that is functioning and has not been debilitated. A patient with glioblastoma, their P&Ks aren't working anymore. But even with the P&Ks functioning, you do see some cell death. When you look at that with a supercharged natural killer cell, you're seeing over double the killing with an SNK. The presence of NK cell markers in perivascular GBM tissue areas indicates that NK cells can cross a leaky blood-brain barrier and infiltrate the GBM tumors. The supercharged NK cells eliminated the GSLCs in 2D and 3D cellular models in vitro more efficiently than the primary NK cells, which again is this graph on the right. And NK cells secreted cytokines increase in sensitivity of GSLCs to chemotherapeutic drugs like TMZ and cisplatin. Immunotherapy with NKore's SNK cell therapy, NK101, is a very promising therapeutic approach in the treatment of GBM by selectively killing malignant cancer stem-like population and increasing their immune-related surface markers of MHC class I and CD54. Now, I'm going to show you some of our human data that we have now. Our glioblastoma patient data is not fully formed. I did not want to present anything today in our human model that has not been QC'd properly. I will give you some verbal information about what we are seeing in our glioblastoma patient here in just a minute, but this is patient number 1. What is very exciting about patient number 1 is that he came in and he did 9 months of due diligence on our company with his doctors at Mayo Clinic. Jamie is a startup CEO. He has been training other CEOs for over a decade. He is in med tech, predominantly regenerative medicine. He has taken multiple companies public. He has run a multi-$100-million fund out of the University of Florida, where they funded 51 other companies. He is a brilliant entrepreneur that builds companies like ours from the ground up. He is now our CEO after this therapy has brought him back from the brink. When he showed up, we are treating patients currently in Cancun, Mexico, where it is much easier to get into human beings. We are in revenue. I'll talk more about that in a minute. They see cell therapy there like we see tissue here in the U.S. We're going to be using this human data for our IND that we are working on right now. We are very close to having that application ready to be submitted, and we're very close to being ready to taking that first meeting with the FDA. Jamie, as patient 1, we started at extraordinarily low doses. For anyone that understands the natural killer cell field, billions of cells are being given day after day, week after week, month after month. Jamie's very first dose was 191 million cells. This is an extraordinarily tiny dose of cells, but we wanted to gather safety data first, and we didn't want to put anyone at risk, even though we don't see negative side effects from natural killer cell therapies, which is what is so beautiful about this therapy. You're not seeing graft versus host, you're not seeing cytokine storms, you're not seeing this rejection of any kind, you're not seeing the patients get sick or fevers or anything to that degree. We haven't in any of our 500 humanized mouse models or any of the 12 doses we have now given to 5 patients. With this very low dose single infusion that took 3 hours to administer, in 58 days after this gentleman had been stuck at 20% cancer in his bone marrow and in his blood, Mayo Clinic could not get him below that 20% plateau. In 58 days, 75% of his cancer was gone. We saw a major reduction in the tumors in his chest and in his abdomen that shrank anywhere from 21 to 70% in that 58 days. Even more exciting for me as a mom who has watched my child suffer relentlessly from horrific chemo side effects, when he showed up, he was lethargic, he had a horrible back strain, which had nothing to do with his cancer, but was just extremely painful. He needed 4 teas and a nap just to get through the day every day. He was in full body pain, he couldn't work out, he wasn't living. By the time that evening came around, he had already started to feel better. By the next day, he woke up. He had slept through the night for the first time in 3 years. He had energy. He felt these changes happening in his body. His pain had gone away, and that back injury he showed up with was gone. All the inflammation around that injury had subsided, and now he could stand up and walk easily. He was ready to start walking down the beach. By the time he got home 5 days later, his workout routine had fully resumed. And he hadn't been able to do this in 3 years. He worked out 5 days in a row. He felt this good for 3 solid months. We did not give him another infusion until 7 months later. And again, really quickly, here are all of this. Is Mayo Clinic did these scans. This is their report. These are all of the different lymph nodes that he had cancer in, and that is the reduction after 58 days. At one point, Jamie's tumors, when he was first diagnosed, were so large they were 20 millimeters in his body. They had pushed his organs to the side. His aorta was getting shut down. His kidneys were shutting down. He was on the brink when he first started with Mayo Clinic. After the second infusion, we did 350 million cells, again, a baby dose of these cells. And 88 days later, the infiltration of the cancer in his bone marrow was less than 1%. He is now down to 0.023% cancer in his bone marrow with 2 infusions spread 7 months apart with a total of less than 600 million cells. He is now in partial remission. His immune system has been reestablished, and his immune system is holding strong at 21%. His last CT scan he just had several weeks ago, he saw another 50% reduction in those tumors in his abdomen. All the one in his chest are now gone. His tumors, which were once 20 millimeters when he was diagnosed, are down to 5, 6, and 7 millimeters. From 20 centimeters to millimeters. I don't know if I said the centimeters part right first. Started at 20 centimeters, it's now down to 5 millimeters. This is one of the most exciting slides for me to share. This is the first time I've ever shared this slide. This is some new data that we have finalized. Look at this very first graph where you're watching the NK cell count go up as the tumor infiltration goes down. This is a B cell malignancy, and we have seen Jamie's B cell count go from extraordinarily high to now down to an almost perfectly normal range. And you can see on this last graph on the right, the lymph nodes, and how with each dose, they keep going down and down and down. You can see the NK cell counts here down at this bottom grid on the bottom left. He is holding 21.5% And again, he has not had an infusion since July of last year. And then this just shows you the amount of cells he got for the first and second infusion. Then we have my sweet angel, Sophie. She was patient number 2. She went 1 hour after patient number 1. That is how safe these cells are. That is how confident I was my daughter was going to feel amazing after these cells. And this is the picture of my husband and I with the first drop of these cells going in. This was the pinnacle for me. This was climbing Mount Everest for me. This was the moment when all my years of effort, hard work, blood, sweat, and thousands of tears were finally coming to fruition. And we're documenting all of this in a documentary called Saving Sophie, The Road to a Cure. Our filmmakers, they have the show, The Umbrella Academy on Netflix, which is one of the top viewing shows of all time on Netflix. They are producing Kevin Costner's 3-part series, Horizon, and they are filming this for us so that we can use this as not only a fundraising piece, but an advocacy piece. We plan on taking this to film festivals and eventually to the big screen. Sophie's first infusion was even lower than Jamie's at 100 million cells. She actually pulled the IV out a little bit, trying to use the restroom, so we think she really got around 80 million cells. After that, her vision started to improve immediately. Again, she is legally blind. She was supposed to be in pure blackness the first year of life. We used medical cannabis to somehow stave that off. She is a medical miracle when it comes to the vision that she has. But we did see after the second infusion, which was only 141 million cells, we started to see some really exciting necrosis in the tumor. Again, these infusions were spread 7 months apart, and the tumor appears to be crumbling. Her tumor had not shrunk in 5 years on the most toxic chemotherapies they had to offer my child. Her last 12 months in treatment, she was so sick on 4 chemotherapies called TPCV, she could not even go to school. She's never been healthier. Her vision is now better than it has been in 7 years. She just recently got 2 additional infusions. Actually, sorry, let me back up to this one. She got 550 million cells and then 1 billion cells spread 30 days apart. This 1 billion infusion was given about 2.5 weeks ago. She gets her next scan on October 13th. We are very excited. These are the biggest doses we have given this close together to any patient, and she continues to improve clinically. So we're really excited to see what's going to happen to this tumor in October when we get that next scan. This is what my daughter looked like on chemotherapy. She was sick. She was frail. She was skin and bones. We could see her spine. We could see her ribs. Her hair was thinning. For years, we did not even know what her natural hair color was going to be. We expected it to be blonde because I'm a natural blonde, but it was white. This is not my daughter's natural hair color. These drugs are so toxic and so debilitating, and the long-term effects that she's had from this, like ulcers in her gut that she's now on 3 medications for. She has extreme anxiety and OCD from all the years of torture that she's been put through. We had to add a second medication to help her with that. This is not what we should be doing to human beings. It is absolutely ruining people's lives. It is destroying their families. It is destroying their communities. We have spent over $1 million out of pocket trying to save our daughter's life, and it has absolutely devastated our bank account. This is what my daughter looks like on natural killer cells. She is healthy. Her hair color, we know what it looks like now. She is a strawberry blonde like her mother. She is a public speaker, who you got to see just a few days ago. This kid has more energy than any child you can even possibly imagine. Thankfully, she is not shy. Cancer has not stolen her sparkle. She is a regular advocate on DC. She speaks for the Congressional Pediatric Cancer Caucus every year. She goes door knocking, advocating for children with cancer. Sadly, this year we did have 100 out of 160 million in our grant funding for pediatric cancer taken. A lot of the grants, as you know, have been taken as well. So we are all in a very, very difficult industry right now. It is very hard to close capital. We are actively raising $20 million. We have gotten as far as we have on $4.5 million. We have manufacturing set up in the United States. We have manufacturing set up in Cancun, Mexico. These are the 5 patients we've treated. These are all of the doses that we have given. We just recently, last week, treated a recurrent testicular teratoma. We have cleared a stage 4 breast cancer patient and another second glioblastoma patient. They are both being treated the second week of September. The first glioblastoma patient that we treated is now been with us for over 20 months. She is in vacation in Italy. She does have some neurological issues that were created by this tumor. Her disease had already crossed the midline when she came to us. Once glioblastoma crosses the midline, you're not coming back from that. She had lost vocal function and use of her right hand. After her last infusion, she got the full use of her right hand back. She was knitting, she was gardening, she started speaking in full sentences for the first time. She then took off on vacation. Sadly. Good for her, not great for our data, but this is medical tourism, and so we cannot control what our patients do. Let her live. We always wanted to prove life extension with her regardless. We always wanted to save her life. We never expected to save her life. There still is a possibility she will beat this disease, but without being able to give her regular treatments, we don't have as much hope because this tumor is so aggressive and it was already so far past that early stage diagnosis, but we will see. Hopefully when she comes back in September, we'll be able to give her an additional infusion. She will be getting another MRI at that time. The edema in her brain that we have seen a reduction in has been astronomical. It is phenomenal. We have seen one of the areas of disease shrink and then stabilize with a very, very low dose. And we have seen another area, we believe it's pseudoprogression, but we're not sure until we get that next scan, despite the fact she is still living. And this is still very exciting. So currently, again, we are raising $20 million. That will get us through our IND approval into phase 1 trials. We believe that this therapy, with its no toxicity, we believe it's going to be a more effective cellular therapy. It is off-the-shelf allogeneic. We can cryopreserve this and ship it anywhere in the world. We are in talks right now with the Medical Tourism Association. They are ready to onboard us and give us some very exciting technology for free that's normally $25,000. They have the ability to help us set up clinics all over the world. We got a personal introduction from the global CMO at MasterCard, who is a dear friend of mine and has been following my journey for over a decade now. With the right funding, we are ready to run. We are going to continue self-funding. We have put a $200,000 investment into our manufacturing, which is going to get us 8 runs. That will treat over 15 patients. It's going to depend on how many cells we get out of each run. We hope to eventually scale that from getting 4 to 10 billion cells per run to 40 billion cells per run, and we are working on licensing a technology that can help us do that. But the team we have built is incredible. Our COO has been through 15 successful FDA approvals. She's only ever had one clinical hold, and it was like a secretarial error of some sort that they got cleared up very quickly and went through approval. And we have 14 rock stars we are ready to onboard today. Usually you only get 1 or 2 of those, but we're attracting some incredible talents, and we are ready to help the people of the world. We believe this is a cancer agnostic therapy. That's why we're treating so many different cancers with one technology. Our animal models, we have treated breast, liver, lung, colorectal, oral, lymphoma, leukemia, stage 4 ovarian, pancreatic, glioblastoma, and low-grade tumors, metastatic diseases as a whole. We have seen the same type of outcomes in all of these tumors in our humanized mouse models. We are seeing efficacy in both brain and blood, solid cancers, and hematologic. Our FDA path is going to be a very broad trial. We already have that clinical trial plan developed. We have Mayo Clinic ready to go, and PI is there ready to run these investigations for us. We have a Mayo Clinic doctor on our board, which is the one that did 9 months of vetting for us, who is very excited to help us drive this ship forward. And it is our goal to create a true paradigm shift in cancer so that patients aren't suffering anymore. We're going to get rid of their cancer, fix their immune system, kill those cancer stem cells, and hopefully then split this company into two after this round and start looking at other immune-related diseases as well. Long COVID, autoimmune disease, prevention, a cancer vaccine that you can get before your NK cells break. It's something we're very excited about developing in the future, but our focus right now is 100% on cancer. Thank you so much for your time and attention.
Story. So we have, let's say 5-10 minutes for questions.
If you get the NK cells.
We have healthy super donors. And what's really cool is that my husband and myself are two of those super donors. So the cells that went into my daughter in patient 1 was my osteoclast that supercharged my husband's NKs. Like I said, we come from the world of medical cannabis. We have done research at UCLA in 27 humans. We have published to a top medical journal proving that cannabinoids have the ability to reboot the natural killer cells and target and kill cancer stem cells that chemo and radiation can't see. My husband and I microdose medical cannabis. I'm 50, he's 51. Our immune systems of that are 20-year-olds. So our immune systems, when put together, it's incredible how beautiful they've worked, but we have additional super donors in Mexico as well that are 20-year-old scientists that we're using, and we'll be working with a blood bank to actually, a New York blood bank is New York Life, New York Blood Bank? Maybe it's just New York Blood Bank. Anyway, they're as big as the Red Cross. So we're looking at a partnership with them in order to be able to get the whole blood that they can then separate the NKs from for us and then just ship it over.
And the doses you were giving, were they based off the dose that they were getting from isolating the NKs?
No, they were based on our preclinical models as far as what we thought would be efficacious. We have now gone through a dose escalation, and because we wanted to prove safety first, right? So now we're doing 350 million to 600 million to a billion. And we're looking at that dose escalation strategy in every patient that we're treating. Each patient commits to 3 infusions. They're $50,000 an infusion. I just billed 3 patients in the last week. We're really ramping up this program, and we've got an incredible way to self-fund, which very few companies at this stage have the ability to do. We kind of did everything in reverse. We got patients treated first, we got it in revenue, and now we're going through IND. But yeah, we're very excited about looking at what this dose escalation model looks like. We want to give as few cells as possible because it makes the treatment more affordable. We believe there is a world where we can get this dose down to under $10,000 a dose. We hope 2,500.
I have another question. So this is kind of interesting. So if this is an allogeneic natural killer cells, you have to, I guess you have to match the MHC complex in any way?
Nope.
Or no?
NK cells are one of those immune cells that do not require a match at all. In fact, a mismatch can actually work better. So there is no typing when it comes to our off-the-shelf therapy and what patient gets it. Any patient can get anything we have frozen.
And then do you expand these cells?
Absolutely.
You have to, okay.
We do that using osteoclast and 9 different strains of probiotic bacteria. It's our proprietary patented process that no one else in the world has.
How many fold of expansion do you do there?
So what, and we are optimizing still, but in the lab, they were taking 1 primary NK and turning it into 5 million supercharged NKs. Now, we have not gotten to that expansion size in the lab. Right now, and I'm not the scientist, I'm not as involved in that part of the process, in the manufacturing process, but we're taking a very small amount of peripheral blood. It's 160 to 100 mLs, and from that, we've gotten as much as 10 billion cells. So when we start apheresing, that's when we're really going to start getting the large volumes, and that's the large-scale manufacturing that we're moving into.
Would you be able to, that's what I wanted to ask you. We will talk later.
Yeah.
Because I will connect you with the largest network of aspirators.
Awesome.
And they get all the...
I would love that. Apheresis is the way to go here. I mean, with apheresis, you don't even really need a ton of donors because you can get so many cells from apheresis. When my daughter actually received IL-2 activated NKs, that were my husband's cells before we did the supercharged NKs, and she got those several, like in 2020. And the amount of cells that they got out of my husband through 1 3-hour apheresis session was insane. It was incredible. So I'm really excited about moving into that larger scale.
And with that, you will might get an investor.
Oh, absolutely. Oh, well, with the people that you know? Oh, love to talk about it. Yeah.
Curious, there's a long time between infusions, so how long do these supercharged natural killer cells last? I mean, how did you figure that out? That I could give them like every year. That's just crazy that they hang around that long.
Well, they're not hanging around. So, and just to be clear, those 7-month spans that we waited in the beginning, that was just to establish safety. Our patients now are getting doses every 21 days.
Okay, that makes
sense. But with that being said, our patient 1, who's our CEO, hasn't had an infusion since July of last year. And his NK cells are at 21%, and they're extraordinarily cytotoxic. And they are still functioning as these incredible supercharged cells. His body is killing his cancer right now. His CT scan just showed another 50% reduction in his cancer. So what and this is, with this next group of patients, what we're doing.
What did he have? What was his name?
Chronic lymphocytic leukemia.
Okay.
CLL. And he had all these tumors in his chest and in his stomach. And then at one point, when he was first diagnosed, he had 90% cancer infiltration in his bone marrow and his blood. It was just all cancer. When we got him, he had 20%, and they couldn't get him below that. Holding steady. It's not going up, and his cancer tumors are going down. What we think is happening, and this is what we're about to prove out with the patients we have now, we think that our supercharged natural killer cells stay around anywhere from 21 to 60 days. We think it's more like that 21 to 30-day mark, which is why we're doing infusions every 21 days right now. These cells come in, they identify the cancer stem cells and any other cells that don't belong in the body, and they start attacking them. But in the meantime, they start training your own cells, right? And they start passing on all of these traits to your own cells. And it's the osteoclast that's making the NK cells behave aggressively, making them so strong and powerful, and then the probiotic is like the superfood to them. So what we're seeing is once our supercharged natural killer cells leave, it is the patient's own immune cells that are now becoming the therapy. We're not changing what the natural killer cells are engineered to do. We're not changing the DNA of the cell. Their main program is to attack and eliminate anything that does not belong in the body, a virus or a stem cell, right? So now it's your own immune cells that are doing the job that they're supposed to do because they've been repaired and they've been boosted. We don't know how long this is going to last for. We know right now in Jamie that it's lasted well over a year with 350 million cells. Now we're giving 1 billion cells. So we're not sure how often we're going to need to reinfuse patients. We need more of that longitudinal data. But my goal, my lifelong dream, would be for everyone at their yearly checkup to get an infusion of these supercharged natural killer cells as a preventative, so we can just wipe cancer out as we know it. That would be my hope.
So what do you do to supercharge these?
Osteoclast and probiotic bacteria.
Oh, just to
Yeah, and we're using a
activate them?
G-Rex.
Activate them?
We do a G-Rex.
Then, I guess since these are allogeneic transplants, doesn't the patient's immune system also react to it in some way?
NK cells are, the signals are very different with NK cells when it comes to rejecting other cells that are brought into the body. It's not like the T cells. T cells, you can have the graft versus host cytokine storm, so forth and so on.
Right.
When NK cells are put into the body, they see them as just natural. They don't see them as something that they need to attack and kill.
Oh, so the patient's
It adopts them.
natural immune system does
Adopts
not recognize.
Doesn't reject them. It recognizes them as part of just it welcomes them in. It's like, welcome to the party. And then it takes on the knowledge of those cells, and then it maintains that knowledge and continues to fight as our supercharged cells leave the body.
Easy. Yeah.
almost like we can all exchange natural killer cells.
Yeah.
Or,
Cannabinoids?
Yeah.
Listen, if I always say that if people could jump in my brain for 5 minutes and just see what I have done with medical cannabis in human beings and the diseases that I have helped to wipe out in patients, I mean, autism. We had kids that had never spoken a word, and in 2 weeks were saying mom and dad. Kids that were latching on to other children with their teeth and not letting go, all the rage is gone. Because cannabinoids, when they go into the body, are anandamide. And with autism, kids with autism have an anandamide deficiency. They're actually using natural killer cells in Cancun to also treat autistic patients. Sorry. We believe there's a very broad application for these cells. We're just hyper-focused right now. But once we close this, we have board approval to raise $20 million right now. We have many investors in due diligence. We have that $20 million circled. It's just everything is taking longer right now. But once we close that $20 million round, this company will split into two, and there'll be two different teams. There'll be a team running the more regenerative side of things, and then there'll be our team running NKore Biotherapeutics. But the investor that comes in today gets both companies. After that, they only get one.
good pitch.
Thank you.
Anyone else? Anybody online?
Interest.
These cells.
Cryogenic.
Oh, you do?
Yeah, we cryopreserve them.
Successfully, and when you saw them, they're like 19.
Yeah, we're getting about anywhere from an 87 to 90% thaw rate right now. We've really perfected our thaw rate, and the cytotoxicity is there. The gamma interferon release is there. They're working beautifully, and at extremely low doses. And I keep highlighting that because that's money.
Yeah.
That's cost.
You.
Exactly. And the more patients that we can treat out of each batch, I mean, it's phenomenal. We can ship these anywhere in the world. The philanthropic angle to this, because I'm a philanthropist, I have my own charity called SavingSophie.org, and my long-term goal, eventually, in 5 to 10 years, is to work with someone like Jeremy Renner, who creates these buses for foundations. He's got a show on Disney called Renovations, and he re-engineers buses. I want to create infusion buses and drop them in third world countries, and I want to give kids free infusions that can't get access to health care, and really use this company to do real good in the world. Getting rich has never been something I cared about. I'd like to have a little money. It'd be nice. But for me, it's about saving my daughter's life, taking away all the pain and suffering that she's endured, and then passing that gift on to the rest of the world.
I wanted to say, you have a very famous predecessor who did that for her daughter, and she is now investing in other companies because she made a brilliant exit, and it was a rare disease.
Who is this?
It's Martine Roseblatt of United Therapeutics. It's a very good story, featured by, not personally, but I did connect with her, was by Tony Robbins. This was the most incredible interview I ever heard.
Tell me her name one more time.
Martine Roseblatt,
Roseblatt,
from
yeah.
United, because she bought a company of a friend of mine, and he sold his artificial kidney. So she bought it, and he left Harvard and is running that for her under United.
United.
That's what you can do with
Therapeutics.
money
Yeah.
when you exit with your money.
Oh, yeah.
She bootstrapped because she founded Sirius XM before that.
Oh, wow.
Oh, wow.
Telecoms.
But she was also the inventor of Siri, the digital radio, and that's where she made her money. And what I wanted to mention to you when you talk with investors, also highlight more your manufacturing and your cost, because you will make the raise if you can really be at a lower cost than anybody else.
Well, I didn't.
You are already in clinic,
Yeah.
so you are already ahead of a few others.
I didn't structure this pitch today for investors. Well, we only set this up on Saturday, and I knew there was going to be a couple of investors possibly in the room, but I brought it in more presentation. Now, I have an investor. We also have those presentations. We have our pro forma budgeted out for all of our Cancun operations and the expansion of that, as well as all the way through to phase 2. We have all of those pro formas figured out. We've got our path to market. We've got our clinical trial plan developed. We have all of those necessary documents that an investor needs to see in order to feel confident in our team.
The reason why I'm saying that is we are right now doing an analysis why none of the cell therapies at CERM ever made it to commercialization. And there are different reasons for that, but one of the main reasons is also the high cost for payers, and that they get niched into certain end-stage patients. Now, of course, these are first generation, second generation. I'm working now with fourth generation.
Right.
Which try to really reduce cost in our allogeneic and broad, as they call it, broad spectrum. Not NKore cells, but.
That's what's so exciting about this is I do think that we can, I mean, it's cost effective now when you compare it to CAR T. A dose of CAR T is like $1.3 million. I mean, we're selling this for $50,000 in infusion, and that's at small scale. The lab we're manufacturing this in right now in Cancun is smaller than this room. So when we get to the larger scale, and we've got like I said, there's a technology out of Florida that's getting 40 billion cells per run. When you start talking about 40 billion cells and an average dose is a billion, and you're at $150,000 a patient, multiply that by 40 and tell me what that math looks like. So, I mean, we've been very, very lean and mean. We're a very lean and mean team now, and to get where we are on $4.5 million, I think is a testament to how gritty we are and how experienced the team is. Most people don't get to revenue and 6 patients treated with $4.5 million.
Gosh.
It's because I don't care about being rich. Honestly, like I've been paid virtually next to nothing compared to what I would normally make in this role, and my CEOs haven't gotten paid anything. The only reason I've gotten paid is because I physically could not survive without it, and I had to sell my cannabis company, which was paying my nice salary at the time, because I couldn't license the patents from UCLA because cannabis is a Schedule I. So,
of course, you want to make money for the investors.
Absolutely. Oh, God, no. We're going to make a ton of money. I'm saying personally, the reason I didn't spend my investors' money was because I didn't care about balloon salaries. I wanted to keep all the money the investors put into the company, so we could get there faster and leaner. And nobody diluted. So they love me for that. We actually have a plan with another investor friend of mine to actually offer our investors now an exit, and they're going to come in with the non-dilutable capital, and then they will make a royalty on the back end, which is a lot like what the Cure Duchenne model has done, where it's very cyclical. It's that non-profit model. So we have an investor that's been a long-term friend of mine that's going to help us implement a model like that into our program, so we can have a lot of equity.
Press the button to keep the light on.
For giving founders more equity as they
cannabis.
Oh, that would be fantastic. We actually have a cannabinoid molecule that we've done research on in our humanized mouse model, and that is what we published to the Cancer Journal, showing the efficacy of that molecule and the ability to reboot natural killer cells and to kill cancer stem cells. One of the things that I'd like to do down the road, again, we're hyper-focused on cancer right now and just the NK cells, but eventually as a tertiary application, we would love to use this cannabinoid. I'm a co-inventor on that patent. We would love to use that as a booster to our natural killer cells, because we showed in our humanized mouse models that cannabinoids boost natural killer cells naturally, along with another probiotic that we have an LOI on out of UCLA that Dr. Jewett also developed. So for us, it's about using non-toxic therapies in a combination or using low-dose toxic therapies in combination with non-toxic therapies to get to that ultimate outcome.
We're invited here, and you're invited by UCSD. I want to put a plug out for UCSD. They have a center which focuses on these cannabinoids for autism.
I've been here many times for those meetings.
Okay.
Yeah,
many times I've been here.
We should definitely connect with also the PI we're working with. got a very much a vested interest in autism.
I'd love to talk to them.
So,
I actually helped start that consortium at UCSD. Yeah.
So you know,
It was many, many years ago,
Yeah.
but the very, very first meetings they ever had here, I was in attendance.
Yeah.
Did a full tour of the campus and, yeah.
And also another shameless, and I'm not working with UCSD, I'm just saying, since we're here, there is a, Professor Ward, former head of nephrology here, built the biggest apheresis center in New York. So it's not a blood bank or anything.
Right, but apheresis.
No, but,
Yeah.
the company I will to with is more like, doing just the trading, with the plasma and the
Wow.
blood. But here is a treatment, it's one of the biggest ones.
You are a useful resource,
excellent.
my friend.
Thank you for joining us.
One other comment, just
Yeah.
might apply for, there's, I'm part of the review committee for CureBound. This is you heard of it? So, basically, they support a lot of basic research, actually, collaboration around.
And they've grants?
They offer grants. That's right,
You have to what?
It has to be in
You
San
have to have
Diego
a
or
little-
investigator in
Yeah.
San Diego with, the UCSD, Stanford, and.
Do they have to be in the universities?
No, not.
No, they just have to live
As long
here.
as you're
But,
San Diego.
future collaborators.
Well, I mean, if it's enough money, I'm sure we can find somebody down here to hire.
Don't you collaborate?
Yeah, exactly.
Yeah.
What they're trying to foster is really, interdisciplinary
Yeah.
collaborations within
Thank
the.
you for that. I made a note. I'll look
Great.
into it.
Okay,
And they're
any
very
other questions?
big into immunotherapy.
I love it.
Yeah.
It's a good space to be in.
What about like compassion? That's one thing that investigator wants to do with compassion. Are they here?
I don't know.
Yeah, you just need a clinician to see what did you.
Okay. We'll have time at dinner. I know some folks have to go, so I
I
wanted to.
unfortunately will not be at dinner, but pleasure meeting everyone. I will follow up on email afterwards.
Sounds
Nice to
good.
meet you.
Great. And anyone else? Thank you, everybody online. I think we might have lost our video, but this concludes our online version, and I'll catch up with everybody afterwards. If you have questions, please feel free to send me an email or follow up. Thanks, everyone.
Thanks, everyone.
Thanks.
Bye.
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 Great presentation.